Good morning. Hi, it feels really early for me and Dr. Wilson, so I'm still caffeinating, but I think almost all of you know me. I'm Allison Gregory. I'm a genetic counselor. I'm from Oregon Health and Science University, and I'll have these two introduce themselves in a moment. So you all had the opportunity to submit questions ahead of time, so I have those, and I've sort of collated them into groups that make sense, but I think some of them were even submitted by people who aren't here because the link ended up in your Facebook group. So I want to make sure we answer your questions. So my thinking is to spend the first 2/3 or so on these and then just open it up and let you raise hands and see if there's something that we have not covered. The other maybe obvious thing to some of you is that there are a few people missing up here this morning who are usually here, and that's Dr. Hayflick and Dr. Hogarth. They're not here, and you can imagine what it would take to keep them away from this. And there's been just a family health problem where they felt they really couldn't be here, particularly Dr. Hayflick. So she sends her regrets. She's thinking about everybody in this room, and we're all going to try to represent them as best we can, but maybe give us a little grace if there's something where I have to say, I don't know, but I will get back to you, because that might happen. So I will let Dr. Wilson and Dr. Thakur introduce themselves, and then we can get started.
Hello, I'm Jenny Wilson. I'm a pediatric neurologist also at Oregon Health and Science University, and I see kids with MBIA disorders and also more broadly kids with neurodevelopmental differences and general pediatric neurology.
Hi, I'm Nivedita Thakur. I'm from UT Health. I'm a pediatric neurologist, movement disorder specialist, and I work with kids in the MBIA community, but also in a lot of rare neurodegenerative diseases. So thank you for having me here today.
Okay, so sort of on our docket, when I looked at the questions that were submitted, they fell into a few different categories. One was gene therapy. One was our CoAZ product. Many of you were in the trial for that and sort of what's happening there. We had some clinical questions and some just more general questions. So I'm going to start with a few softballs to get us going with some of the clinical ones. So maybe Jenny and or Nivi can address. One of the questions submitted was, how do we monitor effects of PKAN on the retina, or maybe just eye health in general?
I think most important, and I think that's something that hopefully gets stressed when you are being followed for PKAN, is to have an ophthalmologist that is following. Oftentimes, getting an ophthalmological exam can be challenging in the little ones. So, depending on when the diagnosis is made or there's concern, I think it's important to establish with somebody who has experience, and sometimes you may not have someone who has experience specifically in PKAN, but understanding the effects of what happens to the eye in relation to neurodegenerative conditions is important. And so if you have someone who has that understanding, they can keep a close eye on the overall health of the eye and retina in particular. And I don't know if Jenny, you want to?
Yeah, I mean, I think it's also important to know that we don't have a specific treatment for the pigmentary retinal changes that happen. And so while, yes, you can monitor those changes periodically, you just have to be aware that there aren't any specific interventions based on those exams. And so in general, we're not recommending repeated sedated exams to just document. So I think having a regular ophthalmologist to check in is great, but in general, if they're recommending a sedated exam, you should be asking, well, do we need to do this? Is this going to change management?
Yeah, and I think the other word you might hear in there is electroretinogram, or ERG. That would be maybe the sedated one, or the one that takes more time, where again, it could give you information but not necessarily change management. Okay, another one that I think both of these two can address, but maybe speak to kind of more generally, because this is going to vary from child to child. What accommodations do you recommend for school services? Or maybe what things should even be considered? Like, what should the thinking be around that?
Yeah, that's a very broad question, and it takes a probably individual answer for each child. So probably everybody with PKAN, every child should have modifications in their school services and individualized education plan. But it's honestly probably going to look different for everybody. And I think where each child is in their disease, it's also going to change over time. So some kids don't, need to stop attending school at a certain point of time, or decrease school participation if they're severely affected. Some kids don't, will be fully participatory, but might need, additional supports for communication, occupational therapy, physical therapy, things like that. I think making sure that the child's physical therapy, OT, speech team privately has input, communicates with the school as well. And sometimes it's helpful for the physician to write a note or to provide input to those IEP meetings, so each child is getting everything that they do need. And particularly, kids with PKAN often are going to have normal cognitive function, but have challenges with communication and motor function, and we really want to make sure they're accessing their education fully. And that may take creativity and really good therapists to help achieve, but that may be in the regular classroom, and for some kids, maybe not for all kids, but it's really going to be pretty individual. And I think as a parent, often you're the main advocate there, but use your therapists who know your child best to help with that advocacy. And if you're not getting what you need, there are ways to people who can help you advocate further to get those things.
I think the most important thing is if you can kind of, like Jenny said, have your therapists that are working with your child understand what they're seeing and what they think would be beneficial, and then having that be communicated to the school. Ideally, I think communication is the key. I think the schools can, see your child at one point in time, but knowing what are some things they need to look out for, what are things that are going to be helpful to help them be the most productive and enjoy going to school, I think is important. So that communication between the therapists that are working locally with you all and the school together, and then coming up with an IEP plan that's really reflective of what the potential and what are some things that are going to be important to be addressed. Make sure those are getting addressed.
Thank you. And then the other clinical question that was submitted ahead of time, which I know Dr. Wilson can comment on because she did a little study on this years ago. Do you recommend THC, CBD for symptom management? And maybe how does that play in with other medications, and when you would think about that?
Yeah, so I think CBD has been well studied for epilepsy. THC, not very well studied, particularly in children. And so, using a combination or a product with THC, we're not using it based on good data, let's just say. In adults, we know that the combination of CBD and THC for adults with, for example, multiple sclerosis can be helpful for their muscle spasms, but the studies there even aren't that great. So we don't have any data to guide this. We have experience, and the CBD component probably doesn't do anything alone or very much alone for dystonia or pain. It's fairly safe. There aren't a lot of adverse effects from CBD alone, but it may not do much for your child or for your loved one with PKAN. So if you're going to use something that you might actually have efficacy, it probably would have to contain THC, but we don't know if it actually works. And there are a lot of downsides to THC, particularly on the developing brain. So we have to use a lot of caution. In general, if kids are not in pain, they're not suffering, their dystonia is not severe, I would not recommend using CBD THC. It's only when kids are really suffering, I would say, with their dystonia, having a lot of pain, and our traditional approaches are not cutting it, where it might be reasonable to consider these products. And having looked at this and asked a bunch of you guys if they're being used, yes, people are using THC or CBD and THC with their loved one with PKAN. So, but we just don't know how well it works. And the kids I've seen who've used it have had some benefit from it. But it's really, we're using it when the benefits potentially would outweigh the downsides. And again, THC is the part that makes you high. And so if you're giving it to your child, they might get a little bit high, and it might be affecting their brain development. So it really has to be kind of worth it in that regard. So really the kids who are struggling a lot with their dystonia, you might want to think about those things.
Yes.
Question about the form of THC or CBD that you've seen in the clinic being used. I think in Oregon maybe it's a little different than other parts of the country, but is it an oral kind of gummy kind of form that's normally used? And then comment on CBD bioavailability. It's not taken up by the oral route very well, and so it's a challenge to even know CBD works because so little is getting in systemically to the body.
Often people are using oils. I mean, I think gummies are an option, but usually it's kind of an oil. I know with CBD that's been fairly well studied, giving it orally to children with epilepsy, and gets into the body, the blood, the brain, and is effective. I don't know how well that's been looked at for THC. I don't think anybody gives it to a child, has them smoke it or anything like that. I think it's mostly oils that people are using. But these, you can't be prescribed. This can't be prescribed because of it's a drug basically. So a physician cannot prescribe it. If it's legal in your state, sometimes a physician can sign for a medical marijuana card for a child, but that just leaves the parent to basically go to a dispensary and fend for themselves, trying to figure out what product to get. And those are not well regulated or regulated at all. So that's a big challenge, and they're not cheap either. So if you're gonna kind of be giving this therapy to your child, it's a big expense, lack of regulation, and these are many of the downsides, I think, with this therapy.
Do you know what kind of THC has been working with liver dystonia? Or people been using the exact one, the exact dose?
There are like a bajillion different products and strains and all that, and so not really, yeah. I usually connect one parent to another parent.
That'd be great.
Okay. I'm going to shift a little bit to an update on our CoAZ product, and I know a lot of you are probably interested in that. I think I'm the only one sitting up here who's been directly involved in that, so I'm going to do my best to be Susan and Penny and talk about this for a minute, but know that I could talk about it for 2 hours, and I obviously can't do that here, so I'm going to try to keep it short, and then if there are a few questions, that would be fine, but maybe it's also a conversation for outside this room if there are other specific questions. So some people asked what the results of the trial are. So what I can tell you is we have not yet published any results from that trial. We have a manuscript that I and a few other people have been working on. And it's nice to have something submitted and accepted before you start talking too much about all these things. But in this room where you all are participants, we're happy to go into this. We have only analyzed the data from the first 6 months. So the way the trial was set up, it was 6 months of what we call randomized, controlled, double-blind, with the placebo, where nobody knew which group they were in. There were 3 dosing groups, what we call low, medium, and high dose of CoAZ, and then a placebo group. So from a scientific perspective, that's the most important time period that we wanted to analyze first, were those first 6 months. And we have looked at that data. We've looked at the biomarker data from that. And I can tell you that we saw a beautiful relationship that made our statistician, want to jump out of her shoes, because you don't see this all the time, but a very clear dose-response relationship where the higher the dose was, the more response we saw in the CoAZ mRNA product that we look at, which is sort of an indicator that it was doing something, that it was working. So, like the low dose line was here, medium dose and high dose. So that's basically, if you're an investigator, that's what you want to see. It's sort of amazing. So we were really encouraged by those results. That's part of the manuscript that we're working on now. The other thing I should remind you about the trial was that biomarker that we looked at was sort of a secondary outcome measure that we looked at. This was primarily a safety and tolerability trial, meaning when we gave it to people with PKAN, was it safe for them? Did it cause any side effects that were serious or not so serious? So we have looked at that data in detail as well for those first 6 months, and we've been really encouraged that it appears to be very safe. There were no serious, what we call adverse events that were related to it. We didn't see a difference between the 3 dosing groups in the number of tummy aches, headaches, different things that we tracked going on during those 6 months. And we didn't see a difference between those 3 groups and the placebo group. So that tells us that it was well tolerated, that it didn't cause any side effects that we were particularly concerned about or safety problems. So I think that's all really encouraging. The collation of all that data is what we submitted to the FDA, which was the next question that multiple people submitted. So we submitted what's called a food additive petition to the FDA in February. Some of you are probably already familiar with this concept. It's been in the newsletter in a few different places, but we, meaning the grand we, more Susan and Penny making this decision than the rest of us, did not want to submit this as a drug. We wanted to submit it as what's called a food additive, because then we can make what's called a medical food. And a medical food follows a completely different pathway through the FDA than a drug, and it's a pathway that's easier to navigate and usually faster, and the requirements are a little bit less. And because CoAZ is something that occurs naturally, it's in our food in small amounts, it's pretty easy to make an argument, especially with our safety data, that it could be considered a food additive and something that was safe to pursue that way. So that means that once it is approved, and I don't know when exactly that will be, then we can develop it as a medical food, which won't require a prescription per se. Like, people who take a medical food should still be monitored by a doctor, but the bar, again, is lower in terms of requiring a prescription or that sort of thing. I know everybody wants to know about timing. I mean, the FDA, once you submit a petition like that, they say they have 6 to 18 months to respond. And with the political climate right now and everything going on, I have no idea if it's going to go faster or more slowly through the FDA. I think it's 50-50 at this point. So we're hopeful, but so far they've been really responsive. So we've taken that as a really good sign that the FDA, the minute we submitted it, we got confirmation. They've communicated with us very well all along. So some people have asked us, should we be signing petitions? Should we be going to our congresspeople? Should we be banging down their door saying, do this now? For now, we kind of want to sit back and let them do their work, because it hasn't been very long. And they've been very responsive. And then if we get to a time point where we're like, this feels like it's taking too long, we might call you all in to start banging down their door a little bit. Yeah. Let's see if there's anything else related to this. The other questions submitted were more related to what happens after it's approved in terms of manufacturing, distribution, that sort of thing. That's pretty far out from our perspective, so I don't have a lot of answers regarding those questions. What I can tell you is it's incredibly expensive to make a tiny little batch. And by expensive, I mean like $500,000. And it has a shelf life. So what we don't want to do is get ahead of ourselves and pay $500,000 of your hard, raised money that you've donated to Spoonbill or other places to manufacture something that's going to expire before it's approved, if that makes sense. So we're keeping an eye on the situation. Obviously, we're thinking about it constantly in real time and how the timing of that might shake out, but we're not currently manufacturing it because we don't know when it will be approved, and that's a pretty big spectrum of time where that could happen. So I'm talking a lot. What questions do people have about that? Yes.
Can me the difference between a medical food and a dietary supplement?
I can try, and these guys might need to help me. I mean, a medical food is a separate designation. So like disorders like PKU, phenylketonuria, there are certain medical foods that don't have phenylalanine. There are other rare genetic disorders with medical foods. I mean, a supplement is less regulated, is my understanding. A medical food still has a bar. The bar is just not quite as high as a drug, but supplements, I think, are over here with very little regulation.
Yeah, I think dietary supplements aren't regulated really by the FDA at all, and you don't have to prove that they can do anything, essentially. But, yeah.
Is the active component available in dietary supplement form, or it has to be put in the food in order to be available?
It's not available at all right now. So the chemical name is 4-phosphopantothene, and it's something that we, over the course of 10 years, again, the grand we, not me, because I'm not in the lab, literally designed, built in a tiny little quantity at OHSU with our core people there, transferred to a manufacturing company, that had to take 2 years to learn how to make it. So no, it is not available anywhere else. Martha Nell?
Is it going to be effective for atypical as well as the PKAN?
So effectiveness is a question that we haven't tried specifically to address yet, which I know sounds crazy, but like I said, this was a safety and tolerability trial, because that's really the first step whenever you develop something like this. Before you worry about whether it works, you have to worry about whether it's safe. So that's the data that we're looking at and looking to publish now. We do have secondary data, because all of you were in PKAN ready on your symptoms, and eventually we hope to look at that in reference to when you were in the CoAZ trial and what dosing group you were in and all of that. But we don't want that to slow us down on getting it out to people, because it is safe, so we want to get it out there so people can try. In terms of effectiveness, we have anecdotal data. We have comments from you all about, yeah, I think my balance feels a little better, my speech, that kind of thing. In our perfect world, we would catch every baby with PKAN the minute they were born and start giving it to them then. That's when we think it would work best. That right now is not realistic, so I think it's going to be individual in terms of what it, where people are in their disease and what it might or might not be able to do for them is going to be an ongoing learning process.
And how will you determine that dosage? It won't be the same for everybody, but with us being so scattered, how will our neurologists know?
Yeah, the dosage will probably, so we did it by what's called body surface area, which is a combination of, height and weight. So my guess is it'll be something like that. So that's how we did it in the trial, and there was a low, medium, and a high dose. They were all determined to be safe, so I'm guessing we might go with the higher one. And then it'll be calculated based on your size. And again, like if a child is growing, that means it's gonna have to be updated just like we updated it over the course of the trial for younger kids. Della?
You might not be able to answer this either. So you had said if you could give it to an infant when they were first born. So if someone's diagnosed at 3, that if you could start the drug at 3, they probably would not have the deterioration. That's your hope?
That is our hope and our dream. We have no idea if that is true.
Okay.
Yeah. We did have one pretty young child in the trial who was a sibling of another person with PKAN, so we were able to catch that person young. But we only had, 18 months. So we just, there are a lot of unanswered questions. What we don't want is for all those unanswered questions to stop us from getting it out to everybody because it's so safe. Yeah.
Okay.
Is the plan to conduct additional clinical trials with this compound, what you call phase 2 or 3 type clinical trials? Or you just going to get it out there to everybody?
I think we just want to, the priority right now is to get it out there as a medical food. And part of that is fun. So our first trial was funded by the NIH, and it took us several years to secure that funding. To go for funding like that again takes years, the way their cycle works. Or you can fund it privately, which means more fundraising by you all and other philanthropic groups. So, yes, ideally, we have this natural history study established. It would be pretty easy to know who's on CoAZ and keep tracking what's happening with them that way. Is it going to be the same level of rigor as a, you know, drug sponsor-led clinical trial? I'm not sure about that, but we rarely give up the opportunity to collect data if we can, if that makes sense.
Would you provide a journal for parents? I mean, you want to look at outcome measurements of phenotype. How's it working? Just like you said, using message RNA in terms of biomarkers.
Right.
We have a journal, people kind of log when they give it or how often they're giving it, how the age of the child.
Right. So 2 comments on that. One is we did have people keep a journal like that during the trial, and I have these numbers in my head because we've been looking at all of them lately. So 88% of them returned the journals, 12% nothing ever came back. We have no idea what happened. Of the 88%, the good news is most of them were taking their dose 98 to 99% of the time properly, like the right amount and every day. It was a strawberry flavored, you know, liquid. It was pretty delicious and sweet smelling. We're talking about other ways to do it, like a powder that you put in water or a liquid to give it a longer shelf life. In terms of tracking symptoms and that kind of thing, so we do have a natural history study, which is exactly that. It's where we check, you know, it's a IRB-approved natural history study. It's been going on for more than 10 years. Many of you were in it. And we do updates every 6 months where we're asking about specific symptoms and tracking and new changes, that sort of thing. So I think it would probably best be done that way so that it's consistent and it's IRB approved and, you know, a proper research protocol rather than just everybody kind of keeping a journal at home. But there's nothing wrong with keeping a journal at home. It helps you with those study visits.
But sometimes helping people, when they have a journal, the categories to look at, these are the outcomes we
I
want to
agree.
look at.
Yeah, I agree.
Subjective, it's better than.
That's good feedback. Okay. I'm going to move on. The other category that we had several questions submitted for was gene therapy, what's happening, updates. And I wish Susan were here because she could talk about this in detail as well, but I'm going to try with everybody else up here to address this a little bit. As many of you know, including Shireen, who's in the room, we've had a huge movement towards both fundraising and developing a gene therapy for PKAN. We're doing that at OHSU in partnership with UMass. They have a Center for Gene Therapy in Boston, and we have some really good partners there who have deep experience with doing gene therapy and other disorders. We did not have that deep experience, so we needed a partner who we could work with. We know PKAN inside out, they know gene therapy inside out, so when you put all of us together, we cover all the bases pretty well. There are questions about what we've seen so far. So I can tell you where we are with the animal studies, and then maybe Jenny can comment on what we've been talking about, you know, in terms of developing a protocol for kids and adults. So some of you may remember we have what we call a colony of PKAN mice. We've had it for many, many years now. And the mice genetically have PKAN. They don't have the same symptoms that people with PKAN do, although that's been a frustration that the mice don't have the same symptoms as people. What Sue Young Zhang in our lab found was if you look hard enough, you can start to find certain signals that do replicate PKAN in humans, including looking at certain biomarkers and expression of different genes in the brain region that corresponds to the globus pallidus, where the eye of the tiger sign sits in people with PKAN. So we've been using our mice initially to just test out the concept, like proof of concept of a gene therapy. When it looked like it was working, we started talking to the people at UMass and developing what experiments should come next. And the next set was looking at how to best build what's called a vector. A vector is like a little circle of DNA. It has the gene, so PANK2, spelled properly and working. And then a promoter, which is like a signal that says, hey, the gene's here, turn me on, turn me off. The promoter's kind of like the dial on a radio to turn it up or down. So one of our next steps was figuring out what promoter to use, because you can pick any number of them or use the one that naturally comes with the gene. So these are all, really detailed little steps that are super important, but I don't know that I need to focus on them here. But, the reason it's taken a few years of mouse studies is that we were looking to figure out the best promoter, how it worked, if the gene was getting expressed the way we wanted it to. We've actually done these studies in the mice where we're injecting the gene therapy right into the corresponding part in their brain to the globus pallidus. We joke around, our lab tech, Helena, we joke about her nursery. I mean, she literally has these massive spreadsheets with all these breeding pairs. How many baby mice do we need on this day? How many do we need 3 months later? And she's literally like planning, and you don't know exactly how many pups they're gonna have or how many are gonna have PKAN, but we call it her nursery. So there's just been a lot of ongoing lab work, which is also expensive because mice are expensive to house and to feed and to take care of during all this. So right now we're on the last step of the mouse studies. We've narrowed it down to one promoter that we prefer. What we wanted to do was see, okay, now if we use this one in the mice, what happens when we look 6 months later and even 9 months later? Is the gene still being expressed robustly? Does it still look as good as it did at 1 month? So that's sort of the last step that we're working on now. And while we're doing that, we're in weekly meetings and talks with UMass and amongst ourselves talking about what a human trial would look like. And I'm going to ask Jenny to talk a little bit about what those discussions have been like, what we're considering outcome measures.
Yeah, so I think people have done this in a number of other diseases, so we're using those models to help guide our thinking with a clinical trial in humans. And we're likely to start with an adult with PKAN for the first treatment, but it seems like we would be able to go to children not long after that we may then be able to do a child and another adult, but the first set of studies is probably going to be a very small number of people, so a handful of people. We need to see how a small handful of people would do after a period of time to make sure there are no major safety issues before we go further. So there'll be an interval of time between each treatment. So one adult gets treated, and this is a injection directly into the brain, into the basal ganglia, monitored for a period of time, make sure there are no safety issues, and then move on to a child. And we don't have everything spelled out in detail. We have not submitted this for approval or anything like that, but just conceptually, that's how we're thinking about things. But there are definitely quite a number of things that need to happen before we get to that point. And then we're looking at, well, how do you tell if it's working? And our primary outcome measure is going to be safety. So that's the first thing we want to prove, is that this is safe. But we'll also be looking at secondary outcome measures of if we can see a signal of, is this working as well, which is a little tricky, I think, when you're dealing with dystonia and the impact of dystonia, because dystonia, as you know, is pretty variable from minute to minute and day to day, and measuring it is challenging. So we're looking at a variety of standardized measures that we might use, and we might ask a few people here in this room to practice some of those things while you're at this meeting, if you're willing.
If we come up to you with a pegboard, you'll know why.
So, but yeah, that's kind of where we're at.
Sarah.
When you look at that efficacy, and I completely understand that it's hard to analyze, but you also look at whether, not that symptoms would necessarily improve, but also if they weren't getting worse, as I think we all see in our PKAN loved ones, is this decrease over time. So is it considered a successful outcome if there's a plateau?
It may be, yeah. I think with a short-term study, we're not going to be able to demonstrate that. I think but the study is going to go on for years and years, like 5-10 years. So ultimately, we will be able to say that, but that it's just going to take time to prove that. But yes, I think that would be considered successful.
I think in the short term too, well, and in the long term, both, the other question is, again, this would be a safety and tolerability trial foremost. So is it safe? And there are immune issues around injecting a vector like this because it travels in a virus. That's how you get it into the cells. A virus that's been, attenuated so that it's not harmful, we hope. But there's a lot of monitoring. Like when we do this, we expect that the person, after they're injected, would stay near the hospital, near our team for probably 2 or 3 months. I mean, it's not going to be an insignificant thing because there are risks around this, and we have to be very mindful of that. Yeah. Yep.
Quick question about, so in gene therapy or things like it, there's a number of different ways to do it. One you described is replacing the gene. You can edit the gene, just a number of other things. So the folks in Boston, they have specific expertise with delivering genes with this AAV, this vector, this virus, and that's why you've chosen this for this disorder. Or is there more to it as far as you're looking at a number of different possibilities, you decide this is the best thing for this particular disorder?
They do have experience with Tay-Sachs, and I can't remember, there's another one that they've worked on quite a bit. We were a good match philosophically, honestly. I mean, we get along really well with them. Our moral compass, everything aligns with them really well, if that makes sense. So we're just all good teammates, and they have good experience with rare neurologic diseases, which is part of it too. They do have experience with brain injections, which you don't need to do for most gene therapies. So to be clear, this isn't a gene therapy where we're trying to incorporate it into the person's genes. It's like you just inject this little vector and it sits in your cells separate from the rest of your genetic information and uses your machinery to do its thing. So that's the concept. Another thing that Amber had asked us to all kind of talk about was, how do you choose that versus, there was another question about this recent CRISPR trial with the child with a rare liver disease. What do we think about? Why can't we do that? Why not gene editing? I mean, the general thing I'll say, and then I'll see what these two would like to say about it as well, is you can approach gene therapy as creating a gene therapy that applies to everybody. Everybody with PKAN has a gene that's not working, PANK2. And ideally, we would develop a therapy that will work for everyone. You're going to spend millions of dollars to develop this thing. Let's make something that works for all. When you start talking about gene editing or ASO therapies that you all have probably heard about, those can be highly effective. They're usually tailored to a person or a very small group of people who share the same genetic change. So part of it is just the philosophy of how you're approaching this. Limitations with funding, there's a lot that goes into thinking around that, but I think conceptually our preference and our goal was to develop a gene therapy that can help everybody with PKAN. You two have experience with these things, please add the...
I was going to say Dr. Adang is here as well, and I think it would be nice to hear a little bit of your experience. I know you're at CHOP and Penn. But I think in general, in the neurodegenerative space, I will tell you, I do some work in Huntington's disease, and I know that it's been very hard in that space to find gene therapy. Again, the whole idea, like in HD, is you're trying to change what's going on in the deep structures of the brain. And so you're trying to deliver something into an area of the brain that is technically challenging. And so I think a lot of work right now has gone on to trying to figure out how to get this into the brain where you need it. And I think that in general, there's been a lot of advances that have been made, and I think right now, for a lot of conditions, they are looking at delivery into the brain. But with that, there's the technical challenges of being able to do that and then kind of looking at what the impact is going to be. So, I think just as a whole, in terms of neurodegenerative diseases and these rare conditions, I think there's a lot of work looking at drug delivery or in this case, gene delivery to the brain. And I think that with some of the advances that are being made, I think we're closer to being able to think about that just across the board, and then obviously in PKAN. But Dr. Adang, I don't know if you have any input. Just I know that study, there's a mic here.
Come say something.
Come say something. I know that story was very powerful, and I know a lot of families are kind of just wondering, but.
Hi.
Hi.
Hi, I'm Lloyd. I'm a child neurologist at Children's Hospital of Philadelphia. We used to have Nivedita, and we actually used to have Jenny too. I think the media didn't get the story right of the gene editing at all. So it was not actually a trial made for the child. It was a trial made to create a platform that could flexibly treat people, where you actually flip out the coding region. And so they created the platform and waited for the child, which is a very different conceptual idea than as was presented. It wasn't made for the child, right? It was made and they waited for the perfect child. And they have FDA approval, and this is where it becomes transformative, to flip out that cassette, right? And so it now is an easy, flexible, fast way to get personalized therapy, right? Overcoming some of the barriers of N of 1, they actually made it a N of 1 plus 1 plus 1 plus 1. Totally missed in all of the media, which I think it's a dangerous road to create treatments for an individual person. I think creating a more flexible, adaptable, faster system is a beautiful idea. And Dr. Ahrens-Nicholas has done a beautiful job pioneering it. I will say, though, we do not have a way to get gene editing into the brain, period. It goes to the liver. And it can potentially go into the blood, but it can't go into the brain yet. I suspect we will be there at some point, but it is so distant I don't even have a guess on how many years, right? It could be 5 years, it could be 10 years. It could be even longer. And I think that that's where it has brought an incredible amount of enthusiasm and excitement, but I do want to temper expectations. We cannot do gene editing in the brain at all.
Can you use intrathecal injections to get around the blood-brain barrier?
No, it doesn't go into the right cells. And I think that's one of the big challenges, honestly, with targeting many of these diseases, that even the vectors that we have that are commercially available don't get in with great efficiency, even with direct injection. And so I think the coming years are going to be us coming up with better, more tailored approaches so we can use less vector, which means it's going to potentially be safer and more efficient and cheaper, right? I think cost is prohibitive for making these therapies widespread.
Haven't they had some success with ASO as far as intrathecal injections and changing in the brain? And that's delivering the message RNA or something similar to that.
Yeah, ASOs work really well with overexpression syndromes, but when you are missing the piece, it doesn't work as well. I think SMA is a quirky alternative because the ASO makes you switch transcripts, but it is not going to be a be-all, end-all fit for these disorders. And also the brain permeability is low, right? You have really dense tissue. You need to be able to get it into the cells. I'm really enthusiastic about it. I work on gene therapy, but I think it is one of those places where we're at the very beginning of the technology. We're at the very beginning of pushing the boundaries of what we can do.
To me, it seemed like ASO could, if it was effective in a certain region of the brain that was important, that it could hold the disease in place, because that has to be continually injected. Because the message RNA has been degraded in place, but waiting for a gene therapy to come along in 5 or 10 years and so on. So, but it's just very expensive. It's a personalized thing.
Yeah, you're right. The cumulative cost and burden of it is high, even though the gene therapies are expensive. They're a one-time treatment. So I think we need to have a reckoning in our biomedical field to make things more accessible and cheaper. And I think that that's where things are going to really take off in the coming 2 or 3 years, is coming up with safer, faster ways to do this. And if we can get more targeted therapies, we can potentially get them cheaper. So.
Yeah, if the government would fund it, that would be helpful. That's another topic.
Cheers to that. I think I'm speaking on that tomorrow.
I'm scared to say this, other questions around gene therapy? No, okay. So we also had some general questions that were submitted. One was about CoAZ, sort of thinking long-term, like should you be talking to your insurance about it, your doctors about it? I would say that's a little premature at this point. I think we need to get approval. We need to, manufacture a new batch, figure out how this is all going to work logistically. But it is something, while it's not prescribed, a medical food, you can get reimbursed. So I think there will be an option for insurance reimbursement for that would be, ideal in our minds. Another question was comment on what's going on in Poland. I don't know if you all keep seeing these posts about all these trials going on in Poland. I don't know. I don't know if either of you know or anyone else in the room. I feel like they've talked a lot about we're doing these trials, but then when you ask them specifically, I haven't gotten specific answers. I think Susan had one conversation with one of their investigators, and it sounded like it was not a drug, but some kind of supplement they were looking at. I don't know if anybody else in the room has more information about that. No, okay, so we're all a little bit in the dark on what's going on in Poland right now, but hopefully there will be more coming soon. There's also a question on the status of Dr. Ben Mamoun's treatment, and does he have a website or place to provide more information? I know you're speaking on Saturday, so you can tell us to save it until then, or if you would like to say something now, you can. Don't want to put you on the spot too much.
I came here to learn, like you. We will be presenting some data on Saturday, I think. There's some promising, exciting news, but there's also a lot of hurdles that we have faced, and we'll be presenting those. I think, don't hold your breath, but maybe the next 6 months we will be presenting the final data. What we have is clearly ready for FDA discussions, but we wanted an additional, basically where we are today, and I don't want to hijack this talk, but where we are today is actually equal or even better than what was approved for the previous studies. But we are investigating one critical thing that we have only discovered recently, and that's why we are going to postpone the discussion with the FDA until we get that, because if that works out, then I think we have something important to push forward. But since I'm speaking, actually I wanted to ask a question here. Is there a way for us scientists to access the genetic data that has been collected over all these years? And I will say why, if that's available to them. Because we've made a very interesting observation, and we can disclose it now, but there might be something that we can extract from the genetic data, knowing the mutations, who's giving the mutation. It's a homozygous mutation. Disease, of course, I understand recessive, but one mutation is probably more severe than the other. Who is the parent giving it to it? And we want that information because we really have something that we obtain in mice, and we want to know that that's actually valid in humans. That might solve another mystery, I think, in this disease, but there's no way to get access to that information. And we thought, if there's a website, or there is some, maybe the associations have access to all the genetic reports from the families. I don't like to contact the families, but there is a way to do it, where you can tell us the allele from which parent, whether it's the mother or the father, will be really extremely helpful information.
So I think there are two answers to that. One is our repository in our natural history study. We have mutation data on individuals with PKAN. Yes, we almost always have that. The question is, do we have the parents? And the answer is sometimes, because the early families, when we were doing actually the gene finding studies, which was linkage back then, we had parental samples and even sometimes SIBs. So if we mined our database, we are going to have a subset where we do have both one or both parents. And then more recently, because they do exome trios, you know, when they're doing these exomes more often where they ask for samples from both parents, we have those in our database as well whenever we can collect them. So our repository, we have the ability to share. We just need, you know, a sharing agreement or an MTA.
That's the issue. If there is, for us, de-identified information.
Yeah, of course. It's de-identified. Yeah.
But MTAs take forever. Every MTA we do takes now 3-6 months, and it's just an absolute nightmare. If there is a way for information like this to be actually shared with the NBA associations through their website, that anybody can analyze, and frankly, in today's technology, probably parents will be extreme help in applying AI technologies to do that type of analysis. So going through MTA, I tell you, for me, it's extremely frustrating because my lab is requested all the time. I have maybe 10 MTAs in progress. They're taking an average of 6 months. It delays science. It's not the best way to move forward. So if there's a way for the biorepositories to actually provide the associations with that information, the associations can remove any medical information, any information about the families, etc. For us, we just need pure genetic, a simple table to tell us this mutation came from from the mother, this mutation came from the father. It will be extremely helpful for us.
Yeah, I mean, that would be almost anonymized, not de-identified, because you wouldn't want to be able to ever link it back.
Yeah.
I mean, no, that's not publicly available right now. You could certainly request it from our repository and we can pull it for you. I don't think it would necessarily require an MTA. We have a sharing agreement for our repository. That's a really easy one-page deal. The other place would be TIRCON. The TIRCON database should have something similar, and I don't know how hard it is for them to
We are
pull.
working with them.
Okay.
They've been very helpful.
Good. Yeah.
Just real, what is he studying? Just, I mean, that, you would just, that's too much? Just a quick sentence as to, not a quick sentence. I'll wait till
Stay tuned until Saturday.
No, we're not talking about it on Saturday, but we are thinking that the severity of disease could be linked to one of the parents. And we want to know that, because if that's the case, then we will know that.
I'm sorry, my dad's behind me and he said it was.
We're not going to start pointing fingers.
No, it will help us. I didn't mean it that way. I meant to say that it will help us pinpoint the pathway that's affected. And that's the information we need. We see it in mice now. We want to know that that's true in humans. That's the main. It's pure experimental exercise.
Okay, so we've got about 30 minutes-ish left, so I'm just going to open it up if there are remaining questions, comments. This is, you know, one of the times. There will be more time on Saturday, but when you all are in a room together, just PKAN.
So if I may, that's why I'm here. I didn't want to interrupt earlier the gene therapy question. Considering or given the high cost of gene therapy studies, given that the technology is advancing so fast, wouldn't it be more appropriate to actually have some form of consortium or at least brainstorming event like during this meeting or others, where actually scientists involved in this study are coming together to think about what is the best approach? Because if we advance the least advanced strategy, it will take money, it will cost a lot of money, it will prevent other studies from moving forward. I know this firsthand because I worked on malaria vaccines, and some terrible studies were advanced. It cost $30 million or so, and that made it impossible, almost impossible for better studies to move forward or to raise the money for them. So my recommendation as a scientist would be actually to bring all scientists together to the table, and we can invite other people from work on the viral particles, work on nanoparticles and others, to participate in the brainstorming session with the hope that we advance forward the best technology that exists, rather than just each little team is doing its attempt, fails, lots of money wasted, you go back to NIH, oh, we already funded 2 or 3 that failed. Maybe it's time, especially for such a small community, such a rare disease, actually to have people come together as a group to try to fight together, rather than each team is fighting on its own. It's only wastes time and money. What do you think?
Yeah, I mean, aren't we meeting this?
We're meeting tomorrow.
Yeah, they put us in a room together.
Yeah.
I don't.
3 to 5.
Well, I don't know that all the parties who need to be there will be there, but I mean, I think it's something that certainly the community wants and makes a lot of sense. And two of the key players are not here today, so obviously Susan and Penny are a big part of that discussion. But setting up something in the future, the near future, for everybody to get together, from my perspective, seems very appropriate and can only lead to good things. So.
At Yale, at least, we try to put together a team for gene therapy, and I can tell you the neurosurgeons we brought together are world class. They performed some successful gene therapies, but communication stopped. And my goal was not only to bring just the neurosurgeons, scientists, but also the scientists who are working currently on some amazing nanoparticle therapies for the direct delivery to the site, to the specific site in the brain. There are no nanoparticles that can go across the blood-brain barrier. We want those people at the table because they just think basic science. It's now an opportunity for them to be part of maybe something translational. And if we don't include them in the discussion, we'll have to wait that 5 years before we learn about it in a publication, versus bringing them on board very early to know what are the limitations of our technology today, what we need, what our needs right now, so that it can help us early on. Tomorrow we'll be attending, but there are a lot of very smart people that we are not inviting to this discussion who can actually be very helpful as well.
Thank you. Yeah, I think that's important to point out. And Shireen, so Shireen is sort of one of the parents who's spearheading the fundraising for this. So would you like to comment a little bit?
Sure, thank you. And I'm excited to meet everyone as this conference progresses. And I would just say, I think that's a brilliant idea, and what I would encourage also is to make sure that parents are at the table too. Parents that are part of the advocacy groups, parents that are doing some of the fundraising, and then parents that are actually experiencing and seeing what's happening with our kids every day. Because sometimes I think, and I'm an attorney, so I
Unfortunately, there isn't a lot of research going on specifically for efficacy of treatments that are already approved for other indications, basically. So we have data from the natural history study, but no study specifically looking at what's the best medication approach to treating PKAN. So it continues to be anecdotal data, using what we know from the literature, from our own experience, and it continues to be a really challenging disorder to treat, as you know. I don't know of any new upcoming or studies looking specifically at kind of the best medication approaches.
Yeah, I think a lot of these medications have been around, and I think what I've learned over time is that different combinations work for different patients. So unfortunately, there's not like an algorithm that I can use to say, even with someone with similar mutations in families too, we see differences in the way that patients respond to treatment. And so, I think that's a little bit of a frustration for families. I think it's a frustration for us as physicians as well when we are trying this, because it is a little bit of trial and error. I think in general, the best approach is that we've got a handful of medications that we know can be helpful, and trying these medicines slowly and kind of systematically to see what that best combination is, I think is usually the ideal approach. I think that there's interesting work going on in terms of different compounds helping for different symptom management. So right now, I know ataxia is not as much of an issue, maybe for PKAN, but let's say, I work with patients that have BPAN, and ataxia is something that, patients can experience. There's actually an interesting compound right now that's being studied in AT that's going through a clinical trial that I think is going to be very interesting in that space. So I think even learning from other conditions and seeing what's being used, and then trying to see if theoretically that could be something that could be helpful for our patients in the NBIA disorder. So that's a lot of how I approach. Sometimes I'm thinking about medications that I'm using in a different condition, but I'm thinking about the reality of, could this be helpful for my patient that has an NBIA disorder? That, and then I will say DBS, again, is something that's very interesting. The technology is getting better. I think there's a lot of work looking at neurostimulation that can happen on its own. That's being studied right now in Parkinson's disease. But I do think that that's going to be an area for us to, look at and learn from, and hopefully be able to apply in the NBI disorders where it's applicable. So that's another thing in terms of technology that I think is getting better, and I think may be helpful for us in the future.
The other thing I'll add to that is, just because I'm on the front line, I'm usually the one who knows who's in the ICU and when and what's going on. Anytime somebody like Hayden or Ben is in the ICU, is struggling, and we hear from them and we set up a call with that team, Susan says we're always learning. We learn from every single one of those calls, and what they tried and what didn't work and what did. And so we have this sort of collective knowledge that continues to grow. And the best practices guideline that we published, I think in 2015, is probably due for an update with some of that, or it's a separate paper. But it's important to get it out of Susan Hayflick and Penny Hogarth's head, and somewhere where other people can access it without calling us, and Jenny too. Because you've been more involved in those calls as well, but we are learning from those. So, well, thank you. That's one thing we try to do, is be available for those kinds of calls. So.
Yeah, I had our therapists have questions. So his PT had asked, if at what point kind of do you call it in terms of therapy? She's kind of asking, will we get to a point that we're okay, now PT is no longer useful, his body cannot handle it anymore? And kind of what we've been told by his neurologist she was just like, you'll know. You'll know when you can't handle it anymore. So is that like a very individualized kind of answer?
Yes, my blanket statement is that there may always be a role for PT. So I think your neurologist is probably right that if you feel like what PT is doing is causing pain, distress, is a burden, then you're probably correct. But I think for some kids there's always a role for PT, and that may be, stretching or giving you things you can do to make your child more comfortable, or helping doing just a little bit of weight bearing, or there may be a role for PT throughout the disease, but for some kids it becomes more of a burden in ways, and you've learned so much already that maybe there's nothing to add at that moment, but doesn't mean you can't revisit it if you say, okay, we're going to pause and then maybe have a one-time visit in 6 months or something like that.
Okay. And then, sorry.
No, go for it.
The OT asked if there's any research related specifically to how PKAN affects the vestibular system.
I don't know of any.
She put in quotes associated with processing, balance, and truncation.
So
Okay.
I will tell you that we know that for a lot of these conditions that the overall sensory profile for kids is different. And so sensory profile, that includes the way that you hear and process, you see, your balance coordination. And so there is a difference that I see in all my kids that I follow, but each one is different in terms of what things tend to be a little bit more overwhelming. For some kids, that vestibular system seems to kind of dysfunction, and so plays a bigger role. For some people, visually, it can be an issue. So I think there's not going to be a paper or something that she's going to find, but I think that what we know is that there are differences there, and so if she can use tools as a therapist to kind of help her identify what are the things that she's seeing in your child that may be playing a role, and then trying to find ways to help address it. And so those are things that you can use across the board. So it doesn't have to be necessarily PKAN specific, but just specific to people who have dysfunction in those areas. That can be really helpful.
She asked about pulmonary function and use of breathing intervention. That's like she's noticed that it helps him, so she didn't know if that was.
What was it specifically that
She says, is there any information regarding pulmonary function and the use of breathing intervention and postural control?
Oh, like breathe, she does.
Yeah, like he does like bubbles and noisemakers and like those kind of things.
I don't know of any specific pulmonary, like the lungs themselves are healthy as far as we know with PKAN, but the breathing can be affected for some kids. Certainly for the kids who are severely affected, the dystonia affects the breathing. It can affect the vocal cords. And even mildly affected kids, how their chest muscles may be tight at times when they're trying to breathe, and certainly impacts their speech. So I think working on those things is very reasonable.
Anything you can do that calms the nervous system down, and we know that there is a direct correlation with, breathing and, changes in breathing. And so if there are things that she's noticing that are helpful in calming, that could be indirectly helping with some of the, symptoms, or even things related to trying to do things like balance and coordination.
Shireen?
For facial dystonia specifically, and I think that's what Loic is struggling with the most right now, what are some things that you would recommend to help with that? I think he's had a lot of hand chewing, and then is Botox an option for something like that? And where can we get that, if it is?
Why don't we let the doctors answer it first, and then we'll.
I think someone may have a suggestion. I think Botox can be helpful. I think especially if there's focal areas that where you're noticing that are particularly problematic. So that is definitely something to look into. I will say usually some PMNR doctors, the physical medicine rehabilitation doctors, some of them are comfortable. Sometimes they're not, and I've seen that some ENT physicians have been able to do because they work kind of in that space. So, depending on it, they may be able to do it. But I don't know, Jenny, if you know, but usually it's PMNR or ENT. I've had some success in trying to look and address whether that's a possibility.
Our PMNR was not comfortable,
Mm-hmm.
and so that was, he said, ask them, and then come back to me.
Yeah, I think ENT may be the other one, depending on where the
Or
dystonia.
if you have good partnership with adult neurology, they often do a lot of craniofacial neck injections. So yeah, I don't know. I mean, that's the best way probably to address problematic dystonia, like lip biting, tongue biting, other than mechanical methods, which also you may be using as well or needing as well. Yeah, there's probably somebody out there, but it could be pediatric neurology, like I do those injections, but it could be somebody in adult might be a good partner. Yeah.
You're in Montana, that's the...
Oh, you're in Montana? Yeah. Oh, I know somebody. Well, I don't know somebody doing injections, but I have connections in Montana, so maybe I can help.
Do you have something to add?
Yeah, they've helped a lot. They've helped, before we started to do injections with him, like he couldn't even swallow water. But we started to do injections in the masters and the muscles right here in the neck. And he's able to swallow water. He's able to kind of swallow like, not as heavy of like a bite, but more without choking. I know that's also like in correlation because he has 2 implant DBSs, but with the injections that we've gotten, we've noticed a lot of less pooling and things like that, and it's become a lot safer to be in.
Caroline, did you have a question?
Oh yeah, I'm sorry. Why do people have a
Why do different... I'm going to restate the question. Why do different people have different presentations of the disease? You're looking at me. So, you all have changes in your PANK2 gene. It's a recessive disorder, so you have not one change, but 2, right? You have 2 copies of the gene. They're both changed or knocked out in some way. And if you think of that gene as an instruction book, like a whole book with chapters and paragraphs and sentences and, words and letters, any number of things can happen in there. You can lose a whole chapter, you can lose a letter, you can lose a word, you can have a sentence duplicated, and the severity of what happens and literally where it happens in the gene all decide how severe that change might be, and therefore how the protein is affected. And it can be in terms of the amount of protein made, the way the protein folds, the way it interacts with some other protein, just any number of things can sort of go wrong along the way. And then you have the combination. You get one from your mom and one from your dad. So all of these things can affect the severity of PKAN, particularly classic versus atypical, but then even within a family, for example. We have families with multiple siblings with PKAN, and they don't look alike necessarily, which just tells you how important all of our other genes are, which we used to say 50 to 100,000. I think now we guess like 20,000 genes we have, but that background is important as well. I don't know what you two want to add to that in terms of, okay. Does that answer your question? Okay. So we're winding down here. I'm going to make a quick plug for the blood draw room. We have a room set up It's right near the lobby, sort of across-ish from where we are now. It is the Palmetto Club room. We have trained phlebotomists in there who are local, and Allison Freed, my coworker, is kind of running the show in there. And we're asking for blood samples only, no skin biopsies this time because Susan's not here. So it's a tube of blood, and we're asking for people with PKAN, but also healthy controls, because you can imagine it's hard to get control samples or samples of blood from healthy kids who are just like, oh yeah, hey, I'd love to give blood, I'm 5. So if you have siblings here who maybe don't mind, we have gummy bears back there, we've got finger puppets, we've got pirate's booty, and all kinds of nice incentives perhaps. But anyone, I think, under 30-ish who might be willing to be a healthy control, or especially young kids like 18 and under would be great, people with PKAN. Most of you have already consented for this, so we're trying to avoid this time doing some crazy paperwork. We managed to pull everything beforehand. We have a spreadsheet. We know who's consented. If you happen to not be, we'll have you do a quick form, but I think a lot of you wouldn't even need to do paperwork for this. And my other request is that we not all go there right now and create a massive line, because you know how that goes. So I would, we're there all day. You're here all day. This session's over, and I think you don't have much else till 4:30 or 5. So if you want to just swing by at some point, maybe a little bit later, if you could do it now. But if you see a line more than 3 people deep, maybe just decide to come back a little bit later. That would be awesome. Any questions related to that? Okay. Anything else you two want to say in closing? I think that concludes our PKAN panel. Okay. Thank you.